Okay, so hello everyone. So let's start the very first Immune Zoom seminar for the new year. So first of all, Happy New Year to everyone. Hopefully 2022 gonna be better, and we have a chance to meet face to face. And today we have a treat, we have Dr. Luke O'Neill from Trinity College, Dublin, gave us a talk about immune metabolism, I believe. So a brief introduction about Neil. So currently, Neil is a professor and chair of biochemistry at School of Biochemistry and Immunology in Trinity College, Dublin, in Ireland. So Neil got his PhD in University of London, and then further move on to become a assistant professor in Trinity College, Dublin, and all the time here until now become full professor. And then 2007, he got appointed to the chair of biochemistry. And then 2011, he became founder director of Trinity Biomedical Science Institute. We know that Luke contributes significantly in the field of immune metabolism. His lab is mainly work on innate immune system, and his lab has discovered a lot of new molecules and biochemical process which are related and triggered by sensors of infection and tissue injury, including toll-like receptors and then inflammasomes. And the signals they drive that stimulate inflammation, particular cytokine in the IL-1 family. His lab also made pioneering discovery in the area of metabolic reprogramming in immunity and the immune metabolism. And also, at the same time, he's using his findings to help the effort to develop new anti-inflammatory drugs. I think Neil is really, I think, talent from my perspective, because he's not only a very good scientist, but also an entrepreneur, because he has founded two startup company. One is Infiltration, and the other is Six Tree. And then he got a lot of award, and I'm going to just list a few of them. 2012, he got Loyal Irish Academy Golden Medal for Life Sciences. 2014, he got European Federation of Immunology Society Medal. And then 2016, he got elected as Fellow of Loyal Society. 2019, he got Landsteiner Award for Biomedical Sciences, Australian Academy of Sciences. I think one thing which I would like to particularly point out is Neil actually also participates a lot of scientific for the public. He's weekly radio slot in Irish National Radio, and he published actually a lot of non-fiction book related to science. For example, 2018, he published Immunology, a Scientist's Guide to Our Amazing Existence. 2020, obviously the pandemic never stopped him. He published another book, Nevermind, Here's the Science, which I find is pretty fascinating. If anyone have the chance, I think you should take a look. And then obviously he gets a lot of pioneer work, published over 260 different papers, and citation over 59,000 times. I think it's really remarkable. So without further ado, the floor is all yours. Welcome, Neil. We're looking for your talk.
Lovely. Thank you very much indeed, Sean. I'm very happy to give the first talk. Who would have thought it would be 2022 already? Isn't it amazing? And this is my first talk of the year. I gave 46 talks on Zoom last year, which must be a record at all kinds of conferences.
I think so, yeah.
Yeah, it's just, and I must say, I'd much rather be there in person. It's not quite the same. Here I am in my office in Dublin. It's lashing rain outside. My lab is just out the door there behind me. So I'm happy to sit in my office and give you this talk. I'm going to give you actually two brand new stories, unpublished, that we're very excited about. They're both going through the ringer in journals at the moment. They'll hopefully come out in 2022. I'm going to give you two brand new stories. One is about JAK1, the kinase JAK1. That's a very important kinase in immunity, as I guess many would know. And we've got an inhibitor, a JAK1, that's based on a metabolite that we're very excited about. And that relates mainly to asthma allergy, actually, as you'll see. M2 macrophages are a target for that, and I'm going to give you the story of that. The second story is about a brand new thing for my lab, and I hope people don't turn off, because it's actually about coagulation. Now, how the hell did my lab end up studying a coagulation and coagulopathy? And it was partly because of COVID, actually, as I'll show you, because as you would all know, I would guess, COVID-19 is a coagulopathy, and many infectious diseases end up causing disseminated intravascular coagulation, for instance. And coagulation has suddenly become an obsession for me. As Tran was saying, I've always worked on innate immunity over the past 30 years, and then the innate process driving the inflammatory process. They're obviously interlinked. And I worked on TLRs for years, for instance, back in the 2000s. And then I worked on inflammasomes, then metabolism, and now I'm working on coagulation. And I hope to convince some of you people listening that coagulation is fascinating. So I'm going to come to that as the second part of my talk. I thought I'd kick off, though, a very general starting point, of course. And my next slide. So this is a review I just did, I think it was in December, actually, last year, with Paul Peter Tack and Chris Buckley. They're both rheumatologists, actually. And we all co-authored this kind of an opinion piece. And it's all about breaking down research silos, is the title, in inflammation research. And we make the case that these immune-mediated inflammatory diseases, even though there's many different diseases, they share common pathways. And therefore, you might be able to block a single pathway to impact on multiple diseases. And we're trying to encourage people to cross-discipline research. And this next slide kind of captures it. Chris Buckley, the rheumatologist, drew this as part of the review. You might have 4 different diseases. It could be rheumatoid, Sjogren's, spondyloarthropathies, IBD, say. And if you're a physician, you might have clinical ways of diagnosing these and using examination. The DAS score is used in rheumatoid, for instance. The PASI score is used in psoriasis. But the truth is, all these diseases, we're now interrogating them from the point of view of, say, digital histology, liquid histology, metabolomics, very importantly, gene signatures, and we're discovering common pathways that all these different diseases share. It's obvious in a way, because obviously you've got macrophages or T cells, or you might have, steroids work across multiple diseases. So we knew kind of there was commonalities anyway, but all the latest fancy technologies that you have to use to get a high impact publication, all those fancy technologies are revealing commonalities between these different diseases. And this next slide kind of captures it, where there's all these shared processes in, say, rheumatoid, colitis, lupus, MS, type 1 diabetes. What are these shared processes? NLRP3, one of my favorite things. CGAS, RIP kinases, JAK-STAT pathways. MicroRNAs control them. My lab worked on microRNAs as well about 10 years ago. The microbiome, the inflammatory reflex is very interesting. The whole idea of tolerance. And then, of course, we're using things like cell atlas, epigenetics, multi-omics, very importantly as well, to inform all this. And the newer things that I think are breaking, sex differences are more and more important. That's a really interesting area. And we know there are sex differences in risk of all these diseases. The stroma, that's Chris Buckley's area, the underlying tissue environment, could be fibroblasts or whatever. They're getting very interesting as well. So all these different things are happening. It's a very exciting time, I guess, in essence, to be working on immune-mediated diseases. And this next slide kind of captures my 4 favorite pathways. And again, the idea here is they're shared amongst multiple diseases. Now, remember, what I'm showing here is 30 years of research. And I'm very heavily involved in the International Cytokine Society. And I remember some of these being described for the first time at those conferences. It's great if you're old like me to watch all these fantastic discoveries emerge in the biomedical world. And it's a great credit to the tens of thousands of us who've done, molecular underpinnings of inflammatory and immune processes, uncovered all this wonder, shall we say. The JAK-STATs are very important, obviously. CGAS, sensing DNA. NLRP3, sensing in the broadest sense, noxious stimuli that macrophages take up. And then I've just got the RIPK pathway here for TNF signaling causing cell death. And these things result in, say, cell death, pyroptosis, IL-1, IL-18, NLRP3. Type 1 interferons are a very important output from CGAS sting. And of course, the JAK-STAT pathway, multiple genes. And then these can go wrong and drive autoimmune disease or autoinflammatory diseases. And you can sort of divide diseases up into those 2 categories in many ways. And of course, companies are trying to make inhibitors. Now, we have several JAK inhibitors approved already. And then there are CGAS inhibitors. NLRP3 inhibitors and RIPK inhibitors in development. And as Tran said, my lab discovered a couple of very potent NLRP3 inhibitors with multiple collaborators, and Roche acquired our company to develop our NLRP3 inhibitors. And let's see what these inhibitors do. I mean, it's a very exciting time clinically to see how these new inhibitors might work in these different diseases. And one very important thing to lead me into metabolism is 3 of these 4 pathways involve mitochondrial dysfunction. And that became clear probably 25 years ago. This begins in many ways, where you see a mitochondrial disturbance linking in to 3 of these 4 pathways, right? And that brought metabolism, I suppose, into the frame more and more. And I did a review, I think about 2 years ago now, with Ivana Mills, one of my great trainees. She did a PhD with me. Ivana's now in Harvard. Great to see the kids leave the nest, and she's got a position in Harvard. It's tremendous. So we did this review together. It's in Nature Reviews Immunology, where the mitochondria become the powerhouse of immunity. And of course, this might be a T cell, it could be an NK cell, it could be a macrophage. Lots of observations about a mitochondrial change linking in to effector functions in those cells has become more and more clear. So this begins actually with cytochrome C release, never forget that, from mitochondria driving apoptosis. And that was discovered by Xiaodong Wang probably 30 years ago now, and that was a real surprise. Here was an electron transport protein doing its job with electron transport and OXPHOS and all that, leaving the mitochondria to very specifically drive a cascade of events culminating in apoptosis. And that begins this adventure from mitochondria to other things outside bioenergetics. And now, of course, mitochondrial DNA has been shown. Toll9 can sense that, driving all our favorite downstream signals and driving immune and inflammatory gene expression. CGAS can also sense mitochondrial DNA, and that will drive the IRFs and drive type 1 interferon. Mitochondrial ROS, reactive oxygen, that can drive MAVs, and MAVs is coming off RIG-I and viral sensing, and MAVs can drive NLRP3, and MAVs also drives its own downstream pathway. And of course, ROS can also affect NLRP3, and that drives IL-1, IL-18, pyroptosis. So in other words, a huge literature has emerged on mitochondrial events, plugging in quite specific ways into downstream pathways that culminate in some kind of immune cell effector function changing based on gene expression. And that's the overarching concept I'm going to be touching on a fair bit today. Now my lab then began to work on metabolic change, and we focused on Krebs cycle of all things. And we kind of stumbled into this. It's about 10 years ago now that we first began doing these experiments. We did metabolomic analysis of macrophages, very important collaboration with the Broad actually, and Ramnik Xavier. He got us into this in some ways, you probably know Ramnik. And we've got into Krebs in more and more detail. And I want to describe for you 2 metabolites coming off Krebs, succinate and itaconate. And my talk is going to mainly be about these 2 metabolites. And what we found was succinate, of all things, again, unexpected. It's in Krebs cycle, obviously, or the TCA cycle, you might call it in America. Succinate is pro-inflammatory, is the bottom line. And loads of literature grew up around succinate as an inflammatory signal. It's got a receptor on macrophages and on neutrophils. It's all these inflammatory events, and we published a fair bit on this over the past 5 years or so. Itaconate is different. It's tied into Krebs. It comes off a conatate. An enzyme called IRG1 makes itaconate. And we published on this in 2018 first, and several labs have published lots of stuff on itaconate now. Bottom line for us is it's anti-inflammatory. So here we have 2 metabolites, unexpectedly, it's in macrophages mainly, 2 metabolites coming off Krebs. One is pro-inflammatory, the other is anti-inflammatory, and that's a fascination. The balance between those 2 then could be determining for the inflammatory process is the overall idea. And for those of you interested in host defense and infectious diseases, there have been a literature on itaconate being antibacterial. Now remember, macrophages make a lot of, in fact, we showed in our 2018 paper, you get millimolar amounts if you put an interferon gamma with LPS onto a macrophage. They churn out itaconate. One reason is to kill bacteria. It inhibits enzymes in metabolic pathways in bacteria. A good example is isocitrate lyase in TB, for instance, and it kills the bacteria. And all these bacteria, TB, cholera, staph, brucella, itaconate can kill is antibacterial. And interestingly, a lab, Alice Prince's lab, had a couple of papers last year on this. Some bacteria use it as a fuel source. So Pseudomonas, especially in the context of cystic fibrosis, actually, they eat itaconate and use it as a fuel source and then limit its anti-inflammatory effects in a clever way. So and that's a really good example of host pathogen interaction. And she's had a really interesting series of papers on bacteria, Pseudomonas rather, using itaconate as a fuel source. And then, of course, there's been a series of papers on an antiviral. And there was evidence it could kill influenza. Very nice immunity paper about a year and a half ago. It can kill Zika virus and now SARS-CoV-2. We can't avoid it, can we? I'm going to show you data about itaconate and SARS-CoV-2 in a few minutes. So again, it seems to have effects on pathogens. That's one reason it's made. But more importantly for us, my lab, we also showed it's anti-inflammatory. And it's got multiple targets in a macrophage. And what we found overall was it can modify cysteines on target proteins in the macrophage. And these proteins that get modified are inclined to be inflammatory and it inactivates them, okay? So we found SDH inhibition. That was shown by another lab initially that got my lab into it, actually, in WashU. SDH, it can target succinate itaconate. It can drive NRF2, a very important anti-inflammatory transcription factor. It can block glycolysis by blocking GAPDH. Now, glycolysis is inclined to be pro-inflammatory. It can drive ATF3, and that can turn on an anti-inflammatory set of genes as well. And then we showed last year NLRP3, one of our favorite proteins, and several labs have shown that now. Itaconate can block NLRP3. Now, to give you that in a single sentence, this metabolite then, it can be antimicrobial by killing bacteria and viruses, but also it can modulate the host side and be anti-inflammatory. In some ways, I think it's a perfect molecule. I'm obsessed with it, so I would say that. Here we have a molecule, a metabolite coming off Krebs. It's able to kill bacteria, say, on the one hand, and suppress inflammation. That could be very interesting as a molecule. And the mechanism of this, by the way, which I'll come on to in great detail in a minute, it's all about cysteine reactivity. So we showed in our paper that itaconate can modify cysteines in target proteins, and then somehow that leads to an inhibition of these proteins. NLRP3, for instance, is a key reactive cysteine. GAPDH is a reactive cysteine. And that led my lab then into cysteine reactivity as a concept, as you'll see shortly. And to broaden that back out for you again, you know, genomics is easy. It's kind of for children. Anybody can do transcriptomics now. We get all these transcripts and then we get gene expression. Of course we do. But what really probably counts in the future is about the proteome and post-translational modification. Massive diversity in the proteome, much more diverse than the transcriptome because of modification. And of course, in the history of biochemistry, phosphorylation was first shown to be one of the modifications. All these modifications have now been reported. Even dopamine and lactate can modify proteins, and that can give rise to all kinds of functional changes, activation, inhibition, degradation if it's ubiquitin. Guess what? Itaconate is another modification. And it was brand new at the time. We had to name it dicarboxypropylation is the chemical name. It's an alkylation reaction, if anybody's a chemist. So here we see itaconate modifying the proteome, and that then will have an output. And again, it's kind of a new idea that a metabolite coming off Krebs would modify the proteome in various ways. And there was a great review, paper just came out in November by Neil Kelleher and colleagues. This is now the new buzz phrase is the proteoform. Now, what's the proteoform? Well, the proteoform is all these different proteins that get made, and they might get modified by phosphorylation, glycosylation, and you get different proteoforms coming off, you know, the genome, I guess. And then, of course, these modified proteins give rise to disease. Say hyperphosphorylation of tau in Alzheimer's is one example, for instance. You know, so suddenly now I think it's interesting if you read that little that paper by Neil. What he's saying is the proteoform is really important, and it's covalent modification, post-translational of proteins that regulate everything really, and that can give rise to disease if that gets aberrant. And it's a very interesting idea. So we're moving towards the proteoform. Now, we've been doing this for 20 odd years, of course we have, but the techniques have got much better to study the proteoform. And itaconate, I think, fits into this because it's another modification of target proteins. Now, let's get into a bit more detail. So why do I say it's anti-inflammatory, itaconate? Well, the target proteins that get modified are pro-inflammatory and they get turned off by itaconate. That's the evidence that we have. But equally, there's more and more evidence like this coming out. This came out about a year and a half ago now, where an unbiased metabolomic screen in plasma from rheumatoid patients identified itaconate in the plasma, and it was inversely correlating with the DAS score I mentioned earlier. So the higher the itaconate, the lower the DAS, and it was inverse to CRP, a well-known marker. Now that suggests it's anti-inflammatory because the less itaconate you have, the more severe the inflammation is. Another paper, this again is about a year and a half ago now, in COVID, where they did a metabolomic analysis in COVID, and guess what they found? Itaconic acid in macrophages was progressively reduced with COVID severity. So the more severe the COVID disease was, the less itaconate. And again, that suggests you're losing an anti-inflammatory effect of itaconate because it goes down. Now that gives rise to a very exciting idea that maybe we could replace the deficient itaconate in diseases and restore balance. In other words, you're kind of, it's a bit like insulin or whatever in diabetes. You're trying to restore something, you know, that was gone. And that makes the therapeutic aspect, which I'll come back to at the end, interesting. But certainly those kinds of papers give us more evidence of itaconate being anti-inflammatory. And it is about cysteine reactivity. And here's my inflammatory protein. There's my cysteine getting modified by itaconate. Now, there's a literature out there of fumarate, another Krebs cycle intermediate modifying cysteines and having effects through cysteine modification. And then on this side, therapeutics have emerged. Dimethyl fumarate is a drug derived from fumarate. It's used to treat multiple sclerosis. People may have heard of it. It's called Tecfidera. And we think that that's leveraging natural fumarate's effects. In other words, DMF modifies cysteines similar to the natural metabolite. And we've made a proto drug that we call 4OI based on itaconate that we think could have therapeutic potential. And I'm going to mainly talk about 4OI as you see in a minute. And again, we've leveraged, if you like, itaconate to make a therapeutic potentially. And it's still early days. This is a proto-therapeutic. But again, the notion here of therapies targeting cysteine to have an anti-inflammatory effect is where my lab is increasingly examining. And try and mention Citrix, our company, conflict of interest, declared they're making 4OI derivatives as possible therapeutics. Now remember, DMF, 3.3 billion sales in 2019. So in other words, if you're concerned about cysteine modification as a concept, that it might have unwanted effects, there's a drug already in people making lots of difference in MS. So again, that notion then of a therapeutic coming out of this, I think is something to keep in our minds. Now, at last, I hear you say he's getting to his data. I'm going to give you two stories now, new stories on itaconate as immunomodulatory and immunometabolite with therapeutic potential. That's my overarching idea here. And the two stories I'm going to tell you about, the first one is JAK1, and the second one is coagulation, immunothrombosis. Let's begin with JAK1. Now, this began with a very talented postdoc in my lab called Mara Runge, who has since left, and she's now doing a postdoc elsewhere. But she had the idea. She came from my lab to work on M2 macrophages, and she spotted a paper. Now, itaconate is made by M1s. It's made by the inflammatory macrophage. M2s don't make it. But there was a paper showing they take up itaconate. So M2s can actually transport itaconate inside. And that made Mara wonder, what would itaconate do to M2 macrophages? Would it modulate them somehow? And again, the idea here is metabolites acting as cytokines kind. Here we have itaconate being made, going into M2s. What would it do? And guess what she found? It was a potent inhibitor of M2 macrophages. Now here's our data. I'm going to mainly show 4OI, our therapeutic. We made that partly because it can cross the membrane with the octyl group. It has similarities but differences to itaconate. But still, we're using it a lot as our tool compound, if you will. And here's IL-4, classic M2 activator, blocked by itaconate, ARG1, FIS1. These are all M2 markers. They're all being suppressed here by 4OI, our derivative. Now, she also tried itaconate itself. And this is IL-13 now, another well-known M2 activator, IL-13, that cytokine. And itaconate can repress MRC1, for instance, in the M2 macrophage. So itaconate can do this. But 4OI is more potent, we think, because it gets across the membrane and gets into the cell. And here we see several markers, IL-13 being blocked by 4OI. Now, what would the mechanism be? Why would itaconate block IL-4 and IL-13 signaling in M2s? Well, they share STAT6, these signaling pathways. And lo and behold, we've got very nice inhibition of phosphostat6 with IL-4. There's IL-4 driving phosphostat6. There's itaconate. There's 4OI suppressing STAT6 phosphorylation. And of course, the upstream kinase is JAK1. And we measured phospho-JAK1 in macrophages. And again, itaconate and 4OI could block phospho-JAK1. So that suggested to us it's acting very close to the receptor here. And of course, we wondered if JAK1 was the target. Now, we also go in vivo. We do loads of in vivo stuff because if you do metabolism in vitro, you can get misled by the medium composition and various issues. So we always try to confirm what we see in vivo. This is a classic in vivo model of M2 macrophage polarization. Judy Allen devised this. It's IL-4C, it's called. It's the IL-4 antibody complex. That will bring out M2 max into the peritoneum. You can then take them out and assay them. And look what happened in vivo. We gave the mice 4OI. It suppressed ARG1, FIS1, and YM1 in the peritoneal macrophages in these mice that we treated with 4OI. So it's nice to see effects in vivo. And then we also blotted for phosphostat-6. That was inhibited again in vivo and phospho-JAK1. Now remember, this is ex vivo analysis, but all the actions happening in vivo. So again, there was evidence in vivo of 4OI blocking M2 polarization was the idea. And then we got at a second model, Phil Hansborough, very important collaborator with Sydney, one of my great old friends. He's a big respiratory expert. He's got the OVA-induced airway hypersensitivity model, which involves type 1 and type 2 immunity in that model, actually, but you get a big M2 involvement in this airway hyperreactivity, and you get massive, you can challenge them with OVA, you get increased airway hyperreactivity. It's dexamethasone insensitive, interestingly, this model, which for the aficionados is important. But here's 4OI, look at this. We're repressing the induction of airway hyperreactivity with 4OI. This is quantified over here. And again, you can measure macrophages in the bowel fluid, and again, they're suppressed. So again, in vivo evidence that 4OI is limiting M2 macrophages in this context in an OVA-induced airway hyperreactivity model. Now, what's the target? Well, obviously, I've given the game away already. We were lucky now because a group in Beijing, Chu Wang's lab, they'd been looking at targets for itaconate using various bioorthogonal probes that they'd made. And we saw in their supplement, JAK1 was modified by itaconate. So I got in touch with Chu. I said, Chu, do you want to collaborate? He said, yes. And we confirmed JAK1 modification in the 293 system where we use this probe to pull out the targets. And lo and behold, 4 cysteines were modified, 2 in the kinase domain and 2 in the pseudokinase domain. There's the mass spec profiles. So JAK1 is modified by itaconate on 4 cysteines was the bottom line. And a second thing we did, another collaborator did an unbiased mass spec screen of macrophages. And they got JAK1. CYS817 was modified. There's KEEP1 in the NRF2 system. We had seen that originally. So here we see JAK1 modification with 4OI, again in macrophages on a cysteine, which is very nice. And then we modeled it. We know a lot's known about JAK family because there's inhibitors. 4 cysteines are right in the catalytic region of this enzyme. So it really does block JAK1 function in that way. And Chu did this for us. This is interesting. So there's a key threonine on JAK1 that coordinates itaconate and we think brings it in to react with the cysteine. So there could be specificity here. Itaconate doesn't modify all cysteine. It's quite specific, and we think it's to do with flanking amino acids. So again, we think that this is quite a specific effect on JAK1 and a few other targets as well. But for the purposes of this, there's specificity with regard to the cysteine modification. And then we also assayed directly in vitro the enzyme activity. It's quite difficult to do this, actually. And here we see JAK1 kinase in vitro, blocked by 4OI and itaconate. They can both block the enzyme activity. Not as good as the classic JAK1 inhibitors, tofacitinib, for instance, but still, we can block JAK1 kinase activity with itaconate, and we think it's got a cysteine modification. Now several JAK family members share these cysteines. We think TYK2 might be inhibited as well, actually. But again, there is specificity here. So in essence, what itaconate and 4OI in particular can do is it can inhibit JAK1. We've discovered another JAK inhibitor, which we think is very interesting. And by the way, the notion of cysteine modification inhibiting JAKs was already there. There's a JAK3 inhibitor called ritlecitinib that modifies a key cysteine and coordinates, again, tyrosines and leucine in this case. And again, that modifies JAK3 and inhibits the enzyme. So that's consistent with what we've seen, cysteine modification in JAK1 being a process. So in essence, what we found then is itaconate can block JAK1. Now, what would that mean? We're following this up in different contexts. As I say, we see in vivo effects, but who knows? We could have come across an immunometabolic link to regulate JAK family members, basically, which could be very interesting down the line in various aspects. Now, my second story, if you enjoyed that one, is even better, I think, to be honest. I'm more excited with this next one. Although that JAK one was good. I'm not trying to downplay it. It was quite nice. But this one is about immunothrombosis. So get ready for a wholly different track now. I'm going to move slightly to illustrate how this is a different story. Now, what is immunothrombosis? Well, the term was coined in 2013 by a group in Munich, actually, in the TUM. And what it is it's coagulation driven by monocytes and neutrophils. Now, we all know blood clots in a test tube. That's fine. You get the coagulation cascade. During infection and during inflammation, you need macrophages and neutrophils to drive coagulation. Why do you need them? Well, the macrophage releases a very important thing called tissue factor. And tissue factor triggers the coagulation cascade. Neutrophils release their nets, and they're assembly points for the coagulation factors. And now we get coagulation, say in the context of an inflammatory site, or indeed during infection, neutrophils and macrophages participate in the coagulation cascade. And it's called immunothrombosis is the name that's been coined for this. Now remember, thrombosis is still the leading cause of death worldwide. 2,000 deaths per day in the US, for instance. Current antithrombotics, there's always a risk of bleeding. So you can give antithrombotics, heparin is given, for instance, but there's a risk of bleeding. There's a need to identify novel strategies that don't compromise normal hemostasis and allow for an anticoagulant effect without causing bleeding. And this is especially important in COVID or in disseminated intravascular coagulation that happens during sepsis, say. There's a big need now to try to limit coagulation in the context of infection without it causing troubling bleeds is the idea. And what we think we've done actually is stumble into a potential way to do this. So keep that in your minds as we go along through this part of it. Now remember, as we're scientists, coagulation is one of the earliest evolved host defense mechanisms in innate immunity. It goes way back through evolution. All these creatures have coagulation, you know. And of course, we ignore it now, don't we? In the textbooks of innate immunity, in that chapter, you will read about coagulation and people forget about it. It's a bit like TLRs came along to reinform innate immunity, I think, in a way. Now, of course, it happens to stop bleeding. Clearly, you want to plug the wound and stop the bleeding. But its second function is to trap pathogens and stop them disseminating. That was shown 80, 90 years ago. And of course, you can trigger inflammation with coagulation factors. So it's very important for host defense because it traps the pathogen and can provoke coagulation as well as preventing bleeding. A really good example of inflammation being driven by coagulation is thrombin. And I hope you all remember, thrombin is at the end of this cascade, drives platelet activation for the coagulation plug to form. Guess what? Thrombin processes pro-IL-1 alpha, and IL-1 alpha, very important cytokine. My lab worked on it for donkey's years, the IL-1 receptor, and we get signaling. You know? Now, we think that might be the actual remnant of the fruit fly system, where Spätzle was shown to drive toll, like a rise to all our great work on the terdomain and the TLR family. Remember, came from the fruit fly originally. Easter is the fly's thrombin, and Spätzle is the fly's IL-1 alpha, and the receptor has a terdomain. So maybe that's the evolutionary conservation there. And remember, thrombin's coming off the coagulation process. So again, a link into inflammation is very important. I'll just show you that as a bit of background because I'm very interested in that for years. Now, how did my lab get involved in coagulation? Well, it begins, it must be 2.5 years ago now, one of my colleagues, Michael Carty in Trinity. I bump into him in the elevator and he says, have you seen these two papers linking inflammasomes to coagulation? And I hadn't seen them. And I read them. They were both in immunity. One was Zhenyu Li, the other was Ben Liu. And what these papers show is a really interesting thing, where tissue factor is the key thing coming out of macrophages, as I mentioned, to drive coagulation. They showed pyroptosis drives tissue factor release, okay? And the two papers combined the following pathway. Caspase 1 or caspase 11, the non-canonical inflammasome, will process gasdermin D, will drive pyroptosis, releasing tissue factor driving coagulation. Now you need type 1 interferons, very importantly, to drive caspase 11 to boost this, what's called the non-canonical inflammasome. The standard canonical inflammasome through NLRP3 will also do this and drive coagulation. Now who would have thought the inflammasome would be able to drive tissue factor release through pyroptosis and promote coagulation? I read those papers avidly because I'm fascinated by inflammasomes and dug out the literature. Ben Liu is the key guy. I've never met Ben, mind you, but I'd love to meet him. He'd some papers. Who would have thought type 1 interferons are driving coagulation? They're doing it through caspase 11, which remember can sense LPS on the cytosol. So that paper shows, of all things, a cytokine ties into coagulation. And the second thing to mention is years ago, and I worked on NF-kappa B back in the late 80s, right? The tissue factor gene was shown to be NF-kappa B dependent, one of the earliest genes actually. And so we think there's two signals here. Signal 1 is to make the tissue factor. Signal 2 is to release it through pyroptosis. And now we get coagulation. And that's emerged as a kind of an idea, a bit like the inflammasome. Signal 1 drives pro-IL-1. Signal 2 is caspase 1 to process it. In the case of tissue factor, signal 1 drives tissue factor production, and now pyroptosis releases it. And all that is in that literature, and it's really fascinating for me. Now, how did my lab get into this? Well, I had a wild idea. Tristram Ryan in my lab is the key person who's going to do most of this. We're going to show he's a graduate student in my lab. And he's brought a huge amount of this project. So I gave it to him and off he went. And it's really interesting what he got. My idea was this. We'd shown in our paper in 2018 that 4OI can block type 1 interferon production. So here we have interferon beta. being suppressed by 4OI, right? ISG20 is interferon downstream gene, again repressed by 4OI. So we knew 4OI could block type 1 interferons. And I just wondered kind of wildly, would 4OI affect caspase 11, which is being induced by type 1 interferon, and would it impact on tissue factor release and maybe affect coagulation? That was the start of this idea. Always ask someone, where did you get your idea? That's where the idea came from. In other words, I was wondering, if we block type 1 interferon with 4OI, we block all these events and stop coagulation. Now, Tristram then said, we won't just work on 4OI, because that's a proto drug and it's a bit of a tool compound. We'll also work on dimethyl fumarate, that I mentioned earlier, DMF. My lab is so obsessed with metabolism. Our lab meeting back in November last year, they made a cake with DMF as a structure on it. Isn't that sad? So I'm going to show you now DMF and 4OI. Remember, DMF is in the clinic for MS. It made $3.3 billion. And because of the overlap with itaconate and cysteine modification, we wondered would DMF and 4OI affect coagulation was the big idea. Now, here's all the data, and it really becomes really interesting. So first of all, there's caspase 11 being induced by LPS through type 1 interferon. DMF and 4OI are blocking induction of caspase 11. They also block pro-IL-1 that we've shown before. Here's quantification of this. So there's caspase 11, there's DMF in green, 4OI in purple. All the slides will have that color coding. There's DMF and 4OI blocking caspase 11. Now that was the start of it. And that was great to see. And then we're wondering now, would that tie into pyroptosis and maybe into coagulation? The next thing we measure is tissue factor, and then it starts to get really novel. So here we see LPS driving tissue factor in a macrophage. Here's DMF and 4OI blocking tissue factor, blocking caspase 11 in the same sample of blocking IL-1 that we've shown before. So here we see tissue factor being inhibited. We then did a big analysis of all the coagulation components in macrophages. And look what happened. They all go up with LPS, the red color. Here's all these coagulation factors. And look at this, DMF can suppress loads of them. So DMF is a very good way to block multiple coagulation factors, including tissue factor being the initiator. So tissue factor is F3 there. So it's really suppressed there by DMF compared to the LPS treated sample. So in other words, DMF is really getting in and modulating many coagulation factors. Now, next then, we begin to collaborate with a lab in Dublin, James O'Donnell, who's a big hematologist who works on coagulation, and Roger Preston, really useful. Oh, my lab didn't work on coagulation, so we went to talk to them and they were really helpful. This is a tissue factor assay, which you can do in A549 cells. And tissue factor is active, and then the activity begins to go away over time, and it's a time course change. And look at this, DMF and 4OI could repress tissue factor activity. This is quantification of tissue factor activity now in cells. Here we see DMF and 4OI blocking tissue factor activity. And then that was in mouse. And then we also did human monocytes. And again, we could block tissue factor activity in human monocytes. And then next thing then we took macrophages, bone marrow macrophages, put on LPS, and you can assay thrombin generation in the macrophage. And you measure a peak of thrombin. And the peak is much faster after LPS. I know it's a bit complicated, but any coagulation lab would know what this is about. So we're getting a big peak of thrombin early with LPS treatment, pre-treat with DMF, and you prevent that shift. So here we see a block and peak thrombin, and there we have 4OI stopping the shift that way. So the orange line is activation of thrombin, and in this case, the purple line is suppressing that activation and pushing the time course out is the idea here. Then we go in vivo. And I'm going to show you now a series of in vivo models. All brand new data. We just got some of this data just before Christmas. This is giving mice LPS in vivo. You can measure thrombin in plasma. You get a big peak of thrombin. And look what's happening. DMF is suppressing thrombin here. And so is 4OI, suppressing thrombin generation in plasma from LPS treated mice. We measure fibrinogen in the mice as well, and D-dimer, they're classic coagulation markers. And again, DMF and 4OI is blocking fibrinogen and D-dimer formation in the plasma of the mice. So it's blocking coagulation, basically. And then you get coagulation in the lungs in this model. It's inhaled LPS, this model. And you can measure deposition of, say, fibrinogen in the lungs. And DMF and 4OI can suppress fibrinogen. There's von Willebrand factor again being suppressed compared to the LPS-treated mice by DMF and 4OI. And then we switch from LPS to live E. coli. So we infected the mice with E. coli. Again, you get a big peak of thrombin in the plasma after E. coli infection, suppressed by DMF and 4OI very nicely. And again, peak thrombin, you can assay it and quantify it. Again, DMF and 4OI is suppressing thrombin after E. coli infection. And then with Rachel McLaughlin in Trinity, one of my great colleagues, she works on Staph aureus. This is a strain called USA 300 LAC. That's a very procoagulant bacteria, well known. And again, we could measure coagulation. There's thrombin after Staph aureus, again being suppressed by DMF and 4OI. Here's tissue factor going up after Staph aureus. Again, tissue factor is being suppressed now in thrombin generation in vivo now with DMF and 4OI after Staph aureus infection. Again, in this model, you get deposition in lungs. This is tissue factor now, massive increase in lungs after this Staph aureus infection. And again, DMF and 4OI suppressing tissue factor deposition in the lungs, which is very nice to see. And then neutrophils, they can make tissue factor. And again, in the LPS model, we're able to repress tissue factor expression in neutrophils in response to LPS. So again, the neutrophil tissue factor now is being blocked by DMF and 4OI as well. We also looked at neutrophil infiltration into lungs in the LPS model. And again, that was suppressed by DMF and 4OI as well. So that was good to see an effect on neutrophils in lungs too. And then finally, we can protect, and this specific bit, we can protect against lethality. So the mice will die of LPS lethality. And look at this, here's DMF and 4OI completely protecting the mice. The caspase 11 knockout is protected, that's known already, we did that as a control. And there's heparin, a classic anticoagulant, preventing lethality in the LPS model. We know the lethality is tied into coagulation basically, and there's DMF and 4OI protecting the mice from the ultimate consequence of coagulation, which is lethality, which is very nice to see. Now, we sent this off then, a referee asked for a therapeutic model. So we gave the mice LPS, we see the clinical score goes up over time, put in DMF and 4OI and we can protect the mice therapeutically after LPS. And that was quite nice to show a therapeutic effect. And in fact, DMF and 4OI again were better than heparin, which is quite nice to see. And caspase 11 were protected as well, obviously, in the model. Now, the last bit is COVID. I was going to show you some data on COVID. Now, remember I said COVID is a coagulopathy. You get massive microvascular coagulation happening. We wanted to examine now the coagulopathy in COVID and see if we could tie in DMF and 4OI into that. First of all, clinical samples from our hospital here. We measured tissue factor and caspase 4 in severe COVID versus mild COVID, and it's elevated. Look at this. And this is in PBMCs from the patients. So we get elevated tissue factor, elevated caspase 4, which is, remember, the human homolog caspase 11. They're both up in severe COVID because it is a coagulopathy. Nobody looked at caspase 4 before. And remember, caspase 4 is going to drive pyroptosis to drive tissue factor. So it was nice to see elevation of caspase 4 in the samples. And then again, with our great collaborator, Phil Hansbrough in Australia, he has the humanized ACE2 mice. He infected them with SARS-CoV-2 and he gave them 4OI in this case. Most of our data is with 4OI here. And guess what happened? It was protective in the mice, in COVID. And here we see the mice developing various clinical scores of severity. There's about 7 metrics, and mainly respiratory disturbance in the mice. And look at this, give them 4OI and we're protecting. So there's none category 3 severity if you give 4OI, and a lot more are category 1 and more are no clinical score. So again, it's protection in a model of COVID-19 with 4OI. And then of course, we also measured loads of stuff in these mice. 4OI is repressing neutrophil infiltration. It's also repressing total lymphocyte counts in the bowel fluid in response to SARS-CoV-2. And interestingly, it's decreasing the viral load. So here we see viral load being suppressed with 4OI in the bowel and also in lung homogenates. So there's an antiviral effect here. Christian Home in our house had shown that 4OI can be antiviral towards SARS-CoV-2. So again, we're seeing an antiviral effect here and an anti-inflammatory effect in the broadest sense and limiting the coagulopathy. And then finally, what Phil's lab has shown here, we're blocking coagulation in the lungs with 4OI, which is very nice. And here's some of the evidence for this. I'll show you von Willebrand factor, it's probably the simplest. So here we see von Willebrand factor, a key indicator of coagulation, deposition in the lungs in response to SARS-CoV-2, and there's 4OI protecting, and here's quantification. So here we see good protection now from coagulopathy in the lungs with 4OI in SARS-CoV-2 infected mice. Now, what does all this mean? Well, I think we've stumbled into a new way to stop coagulation during infection in a potentially safe way. And here's DMF and 4OI, blocking type 1 interferon and also blocking tissue factor. So tissue factor induction is signal 1, DMF and 4OI is working here. And then it's also blocking signal 2, which is the cascade down to pyroptosis. And again, that will block coagulation in an interesting way, I guess. And it's tying in the inflammasome into coagulation, of course. A couple of things to finish up on. Now, guess what? Kate Fitzgerald, another former trainee of mine, had a paper showing that gasdermins can be blocked by fumarate by succination. So a cysteine modification on gasdermin by fumarate can block pyroptosis. And that was interesting because it's like DMF. So it's further evidence that DMF can block pyroptosis, which in our case will stop tissue factor release. And it's all about targeting of cysteines in the end, as I showed earlier, where itaconate and fumarate will modify cysteines and inflammatory proteins. DMF is the mimic of fumarate. A drug called disulfiram, Hao Wu in Harvard showed this, can modify cysteines on gasdermins, and that will block pyroptosis. So again, the notion of cysteine modification therapeutically is really interesting. And then 4OI, our proto drug, modifying NLRP3, modifying JAK1, and then having an anti-inflammatory effect. And in many ways, this is the way to think of it. So DMF came off fumarate, remember, and it's anti-inflammatory. 4OI is coming off itaconate, and it's anti-inflammatory. And there'll be overlaps and differences that might be less toxic than DMF. We do see interesting differences as well. So we think 4OI could be better than DMF, actually, is one notion that we're exploring. And finally, then, the unexpected link from DMF and 4OI to caspase 11, tissue factor, and the coagulopathy. We found a way to block coagulation. And getting back to my earlier stuff, there's a need for new anticoagulants that are safe, is the idea here. And look at this, DMF compared to 4OI, anti-inflammatory, cysteine modification, blocking interference, blocking pyroptosis, blocking gasdermins, stopping lethality. And of course, this is clinically approved and this one isn't. So who knows now? We may get a new therapeutic that will beat DMF is the idea here. And as I said already, our anticoagulant effect is well upstream. It's upstream of tissue factor, you see. The current anticoagulants like heparins are all down here. And guess what? They come at the risk of bleeding. And here's a meta-analysis of sepsis. So if you give a septic patient heparin, there's a big risk of bleeding. You know, this may be a way, because you're blocking well downstream of the cascade, remember, and that might affect bleeding anyway. Whereas if you go early, we think here that might be useful. So could targeting tissue factor or upstream components rather than downstream be safer is the idea here that we're interested in discussing further. And remember, this immunothrombosis business, it's in stroke, it's in infarct, it's in infections, ARDS, and all the rest of it. There are trials already with anti-inflammatories limiting coagulation. The anti-IL-1 trial is a good example. You know, colchicine, methotrexate. There's some evidence of them working for cardiovascular disease. We think they might work in part by limiting coagulation, you know, through the various things I've been describing in a way. And now we want to add DMF to that list. And remember, there's a trial running in COVID with DMF already, the recovery trial. So maybe our data will help inform that trial, because now we add DMF. Now, these people don't know about coagulation with DMF. And we're saying DMF might block the coagulopathy in COVID and therefore have a therapeutic effect. You never know. Now, finally, I want to wrap up with one quick thing for you. I hope you could follow all that. It's hard to do it on Zoom, but I hope you got something out of it. Look at this. So here's itaconate, right? And here's all these different models of inflammation and lupus and all kinds of things. Also in cancer, by the way, I didn't talk much about tumors because it's not my main focus, but there is a literature on itaconate in tumors as well. All these models, guess what? People have tried our drug. We gave away 4OI to many labs. You can buy it now actually from Sigma. And it's working in all these models, amazingly. And these are models of, say, lupus, for example, pulmonary IPF, pulmonary fibrosis as two examples. And now infectious models, our own stuff on SARS, Zika, you know. So all these models, there's effects of 4OI. So getting back to my very start, if you can remember 5 hours ago, all those shared pathways, remember, in multiple diseases, itaconate could be useful then for targeting these common processes that are shared among inflammatory pathologies. And that kind of, again, reveals how, get out of your silo, you know, let's have cross-disease interactions here, because maybe this will have relevance to many different diseases. And it's quite nice to see 4OI then expanding into different models is the idea. So who knows? This could inform mechanistic understandings of disease, of course, through metabolic change. But secondly, therapeutics, who knows, might come out of this, you never know. And I'll finish there and thank the people. Now Tristram, there he is. He did all the work on the coagulation aspects. Mara Runch, there's her name there. I mentioned already, she did the work on JAK1. Lots of collaborators all over the world. Phil Hansborough, really want to mention him. He did the OVA model and the SARS model for us, really important to mention him. Roger Preston, James O'Donnell, Ashling Rehal, very important graduate student in Roger's lab, helped with some of the coagulation assays. Richard Hartley made the 4OI and various other collaborators. And our funding was the European Research Council, the Wellcome, and Science Foundation Ireland. And thank you very much for this.
That was great. I mean, that's covered a lot of ground and a big spectrum. I think we can start from Q&A. If anybody has questions, just please raise your hand or type your question in the chat box. So I guess I'm gonna start to kick off. Actually, I was really fascinated with first part, the itaconate as a metabolite. I'm not familiar with the field, but where it's originated, it's metabolized from the bacterial or?
Yeah, bacteria can make it, and yes, strangely, some bacteria can actually make it. But the main source is macrophages. I mean, there's an enzyme called IRG1, which converts itaconate from Krebs into itaconate, right? And that was an unknown gene for a long time. It was immune response gene number 1. It was named, you know, I think it was 20 years ago. Finally, a guy called Karsten Hiller, very important lab, showed it was the enzyme that can make itaconate. So it seems to be expressed mainly in inflammatory macrophages. You also see it in neutrophils. There's some evidence in NKT cells actually as well. But it's quite restricted. We think the macrophage is the main source. It's a bit like IL-10 in a way, because the macrophage makes to have an anti-inflammatory effect, you know, and then being repressing inflammation. It takes a while to build up, actually, for this to accumulate, but the main source are macrophages.
Right. Okay. So, but in the first part, you show that the 4i inhibit the JAK1, but it seems like system side is relatively conservative across the JAK family.
Yeah.
But you didn't actually go deeper to talk about whether the 4i inhibition also get effective on the other JAK family members. Yeah. Have you tried some of the experiment to verify that?
We have, yeah. No, we tried not a huge amount. We did try JAK3, and that wasn't inhibited, right? TYK2 seemed to be inhibited. So even though there's conserved cysteines, the flanking amino acids are going to be different.
I see.
That threonine I mentioned. So we think there could be specificity here. And by the way, John, if you look at the proteome, we've identified about 50 proteins get modified on cysteine by itaconate. Right. And that's interesting, isn't it? So, and they're mainly inflammatory proteins. So we're very keen on mapping those further, I guess. But JAK1 stands out as a really prominent, as I showed in that unbiased screen, actually. It came up as a massively modified protein. You know? So we think there may be a lot of somewhat specificity anyway towards JAK1.
I see. Related to the second part of the thrombosis part, the DMF inhibition, which actually you show nicely, which experiment related to the macrophage in vitro, but in vivo, yeah. Have you ever tried, let's say, put this way, how you actually tell the specificity of the DMF in vivo? I mean, how you know it's not influenced the other, cell type besides the macrophage?
Yeah, no, you're right. It isn't totally specific for this, obviously. The DMF and 4OY will have multiple targets. Right. But one consequence is that blocking coagulation is the idea, Right. In other words, it's a new way to stop coagulation, as well as everything else. So, for example, John, the recovery trial is in COVID, right? They're giving DMF as an anti-inflammatory there. But now we're saying it's really justified because it will also block the coagulopathy. That's the idea, So there will be multiple things being targeted here. Whereas if you go after thrombin, that's just thrombin, obviously, as a target. The other thing to say, John, is I know it does sound a bit unusual, but DMF is being used a lot therapeutically. It's a weak electrophile, and that's good. You don't want it modifying loads of stuff. And I think what's happening is its weakness is to its benefit. You know what I mean? Therapeutically, and therefore you see a therapeutic effect.
Right, but I mean, in vivo, instead of, or let's say, additional to thrombin, I mean, there's other factors like collagen also can induce thrombosis. And then have you ever, tested the collagen fluctuation during the thrombosis if you treat the mice with DMF?
That's a good thing to do, actually. All we've really done is, remember I showed you the gene expression array that we did, and we saw lots of coagulation factors being repressed, you see. Right. So, but it could well affect things like collagen. We're very keen to do more models, John, to be honest. Right. And there are collagen-induced coagulopathy models, for instance. Nice to try that. I mean, amazing. It's the first time I've shown the data, to be honest. Anyway, so what you've seen there, I know it's very fast. We've E. coli, Staph aureus, and COVID, 3 separate in vivo models, and an LPS challenge in vivo. All 4 we block coagulation, in quite a safe way, and that gives us hope that there could be something in this, basically.
Yeah, but I mean, eventually all the calling factor actually function on the platelets, induce the thrombosis. Actually, I know the DMF is specifically target the factor. I know that the DMF target the macrophage for the macrophage thrombotic factors, but have you ever tried directly treat the DMF on the platelets?
No, that's another good thing. Absolutely. Yeah, that could be interesting too, precisely. Yeah, because obviously platelets are key to this as well, you know.
Right.
There could be a direct effect on platelets, you never know. And it may stop the platelet shape change or the aggregation. So that's a good suggestion to think about.
You know, I'm curious about this because my lab actually started to look into the thrombosis because we're working.
Great.
Yeah, we're working in the serotonin-induced blood coagulation, and that's
great. Yeah. Yeah, I mean, I'm nervous giving it to an immunology audience because immunologists don't think about coagulation anymore.
Right.
I want to get people interested again. That's very nice to hear.
Absolutely. The last thing, actually, I'm curious. I mean, the very last slide you showed, the DMF or 4I actually can function upon various different biological processes. But I'm curious about the functionality of DMF or 4I instead of or in addition to anti-coagulation. But what about antiviral response? Because if you treat the mice with this, so usually there's a complex of accompanied manufacturing with these patients with either thrombosis patient, usually also associated with viral infection. If you treat them with this 4I, do you think it will also influence the drug effect on the antiviral response?
Could, yeah. And in fact, as I mentioned very quickly, there's antiviral effects here as well. So now quite how it's inhibiting viral replication is the question. That was shown with Zika virus, actually, with a neat paper on Zika. And now COVID-2. This guy, Christian Holm, I mentioned him, he's shown a direct antiviral effect. So the question there is actually, is it modifying some cysteine in some viral component, you know, maybe and preventing assembly. And that's something that we're interested in looking at. There's also a very good effect on influenza.
I see.
And again, that's something worth exploring. There's a few labs working on that actually, the viral aspects of taconate, because it's cropped up in a few studies, you know.
But you won't, because obviously from what you tell us today that this DMF treatment actually not going to cause the excessive bleeding or potential risk with bleeding, and that's why even you treat antiviral as a drug, then it won't actually have increased the risk of the potential bleeding. That's the intention of the drug, right?
That's right. Yeah. That's a good point. Yeah.
Cool. Okay, so I see several questions in the chat box. One is from Sarah. So she's asking, there are many good factors of 4I in inflammation or sepsis, COVID disease. So what's the real target molecule of 4I? I think.
Yeah, that's the key question, Sarah, to be honest. Yeah, because we know there's multiple. The challenge here is there's multiple targets, you know.
Right.
I mean, we know it's a good NLRP3 inhibitor, for instance. So we showed that paper on that last year. And as I say, 2 or 3 labs have shown that as well. So the inflammasome is a definite important target, but equally so is JAK1. So it's a new idea in a sense that you're getting multiple proteins being the proteoform. Remember I mentioned earlier, but there's multiple proteins being covalently modified and then they're getting modulated, you know. Mm-hmm. And then the net output is, say, an anti-inflammatory effect in vivo or an anticoagulation effect. That's the ultimate consequence of this. And I always mention DMF because that's safe in humans and it does modify multiple proteins. So in other words, from the therapeutic aspect, we've got to get our head away from a JAK1 inhibitor. It's not that, you know, it's going to target several things.
Yeah, I see. I think the second from Yang. Yang, you want to come out and then ask question yourself?
Yeah, hi, Professor Luke.
Hi.
I'm very curious about the role of 4I on the lung metastasis in terms of any cancer type. Because I see a very nice data on other models, for example, sepsis. It's all about inflammation, and also working by targeting neutrophils. Since we actually study about the LATEs, right? The role of LATEs in lung cancer. So very curious about whether you have ever tasted the role of this 4I on lung cancer.
We never have. That's wide open. That's something you should try because there are a couple of papers on cancer actually with itaconated cells. Dan McVicker actually, one of my dad's on, but he had a nice paper on itaconate being pro-tumor, interestingly. Now whether it's a nutrient or it's being used in some way in tumor cells, we don't know yet. he tied it into tumor-associated macrophage function, actually, being somehow, modulated, and then you get a net pro-tumor effect. So that's only one paper at the moment, but it's well worth considering, because if it does affect any number of targets that might impact on metastases, you could invoke 4OI as a potential anti-tumor agent. So it's worth considering that, but nobody's done that yet.
Thanks.
So I think following Yang's question, I mean, you say there's I will imagine, because I mean the chronic viral infection also always somehow correlate with the cancer development. Do you actually? I would just maybe take a wild guess. Do you think the 4OI's effect during the cancer treatment, not particularly in the lung cancer, in general idea of the cancer treatment, do you think that will actually affect, or the role of I-10 colonate have the effect in the cancer development? What do you think?
Yeah, it's a good hypothesis, to be honest. And as I say, it's underexplored. So there's IRG1 knockout mice, for instance, that can't make any attack.
Right.
They'd be interesting, wouldn't they, to look at models of tumor genesis in, for instance, just to see if they're any different. I didn't show much data on IRG1 knockouts, but we often use those actually as a backup, because they can't make any attackinate, those mice.
Sure.
They get much more severe TB, for instance, because you're losing the anti-inflammatory effect there, Right. They've been put through a few models. Zika, actually, it got worse in the IRG1 knockout as well, because you're getting a big inflammatory reaction. So certainly they'd be worth thinking about in the context of tumors, for that.
I see. Okay. Next question is from Dan. She said, great talk, nice talk. In addition to intracellular accumulation in activated macrophages, I-10 can also be secreted out of the cells. What is the function of secreted I-10?
Yeah, that's another good question. I mean, as I said, I did briefly mention, you can measure it in plasma very easily, actually, And there's 3 or 4 papers now measuring it in plasma in different contexts. Right. The one I showed was a rheumatoid example where it was inversely correlating with disease severity. There's a paper on colitis. You can measure it in plasma in colitis patients. Now, of course, it is getting made by the inflammatory macrophage, so you might see it wherever you see inflammation.
Sure.
And then the question is, what's it doing in that context? I suppose. Yeah. And that's what the question is. Now, as I said, the M2s take it up as one example. So they've got a transporter. We think it's like the succinate transporter is an option there, and they can take it up, and then it starts doing things inside the M2. So it's a very interesting prospect now, and it's not just itaconate. There's evidence for succinate being released, and that has a receptor and gets taken up by certain cell types as well. So you got to think of metabolites kind of like cytokines. I know it's a bit that's overstating it. They're not. I mean, you need a lot of them. They're not very potent like cytokines are, but they are able to affect cells in a paracrine manner, shall we say, and then modulate cell function. The best example we have so far is that M2s will take it up, and you limit M2 function then in that way. And then that was backed up actually by Adam Byrne, who's in Imperial London. He's got lovely data on itaconate preventing fibrosis, actually. And that'll be consistent with modulating M2 max in the context of fibrosis.
How long is half-life of this secreted itinate in the plasma?
Good question, John. Yeah, we don't know any enzymes that can break it down, strangely,
Oh, it's going to stay there.
Well, we've looked for metabolism of itaconate, and it doesn't seem to get broken down. At least so far, we can't find it being broken down anyway. There was a couple of candidate hydrolases that might have done it, but that's another unanswered question. How does it get metabolized? There's a paper I've refereed, actually. It can turn into itaconate-CoA. You can get a CoA derivative. So it's a bit like succinyl-CoA or acetyl-CoA, and that can modify proteins, of course, through itoconylation, and maybe that ultimately then gets degraded through itaconate-CoA production, maybe is one idea people have.
I see. Okay, great. The next question is from YH. You want to ask a question yourself, YH? All right, I'm gonna just read the question. So the good effect of 4I were tested in the immune cells, particularly macrophage. Have you tried to test the function of 4I in non-immune cells like epithelial cells? I think that was one of my
another great suggestion. We'd love you to do that, because no, we haven't. I know labs are looking at it on fibroblasts, mainly in the rheumatoid context, because it is in the rheumatoid joint and when it affects fibroblasts. There's a chance that cells will use it as a fuel source, remember, because lactate gets taken up by fibroblasts in the rheumatoid joint and it's burned through Krebs cycle, actually,
Right.
So you may see metabolic effects as well as all these other effects that we're reporting in a way. But nobody's looked at the epithelium. I'm trying to remember, was the stuff on the epithelium mesenchymal transition? There might have been something in this guy Adam Byrne's paper affecting that maybe. But it's very understudied, that, so it's well worth considering.
Yeah. Okay. You see, I mean, you get bombed with questions. That's usually our museum seminar. There's a lot of questions. Usually, some questions section could be longer than your than your real talk. Okay, I think next one is Zhihua. You want to go ahead, ask questions?
Okay. Yeah. Thank you very much, Chan and Dr. Anil. So I was wondering. Can you hear me?
I can. Yeah, go ahead.
Yeah. Yeah.
Yes. Okay. Sorry. Yeah. So maybe I should turn on my camera. So I was wondering whether the 4OA passed the BBB to affect the function of microglia
It does.
in the brain.
It does, John. The paper on
Zika.
Yeah, the paper on Zika virus. It was about 2 years ago. A big Zika virus lab, actually. I can't remember the guy's name. I must apologize, but he's some Zika lab. They showed it going into the brain. They gave 4OI to the mice, actually. And it was protective in Zika in the brain. Isn't that wonderful?
Oh, okay. Yeah. Okay. Yeah. That's, yeah. Yes.
In fact, they thought that they'd evidence the neurons were making it in the brain as well, by the way. And your own litaconate was having an anti-inflammatory effect on microglia was their idea, you know, and therefore the mice weren't dying of Zika. And if you give 4OI therapeutically to those mice, it got to the brain and was able to save the mice from lethality with Zika, you know. So we do think it crossed the blood-brain barrier. Yeah.
Yeah, thank you. So regarding the immunothrombosis,
yep.
Do macrophage actually interact with platelets?
I think they do. Yeah, that's a great question. I'm learning about coagulation myself, to be honest. But I think they do. Yeah, there's evidence of macrophage platelet interaction. I know macrophages can, and maybe Tran would know more than me, but I know macrophages can modulate platelet function in various ways, you know. So there's stuff going on there as well. Platelets have always been interesting, really, you know.
That's true.
More and more insights coming out of them, in a sense, you know. We'd love to add it to platelets, though. That was a great suggestion to see if it would affect platelet function in some way.
Yeah, so regarding that, you know, if macrophage modifies platelets function, like what about monocytes? When monocytes and platelets, they are, you know, flowing together within the blood vessel, while macrophages probably only meet the platelets when they are disrupted endothelial, right? So I just wonder, do you have any comments regarding
No, but I know monocytes can release tissue factor, you see. So you don't need to be a macrophage to release it. And in the systemic circulation, you might imagine the monocyte gets activated, releases tissue factor, and then that will start the coagulation cascade.
Yeah, yeah.
And then there's neutrophils as well there. And if neutrophils are releasing nets then, and the tissue factor is assembling the coagulation factor, so certainly monocytes are part of this. The stuff that's coming out on is they're depleting monocytes with clodronate, you know, the method you can use to deplete monocytes. And you don't see clotting in response to infection. So that's why they know the monocyte macrophage, if you like. Now, I know it doesn't distinguish them, but that's how we know that cell type is critical for immunothrombosis, because if you deplete them, there's no coagulation. That was using LPS as a model, of course. So again, that literature is growing, you know, the whole time. As far as I can make out anyway, it's getting more and more compelling.
Okay, thank you very much for the fantastic talk. Yeah, okay.
Okay, so next question from Xiaoqing Wei. Go ahead, Xiaoqing, you go ahead, ask question.
Okay, thank you. A great talk. I learned a lot from your talking. Thank you very much. So I just want to ask a small question. We know that macrophage not include only the M1 and the M2 phenotype. So after the lung damage or other tissue damage, the macrophage need to proliferate themselves to maintain their function. So I want to ask this drug 4OI treatment, whether it can affect the macrophage proliferation, especially for the tissue resident macrophage.
Yeah, no, it does. And we looked at alveolar macrophages in some of those models, and the alveolar MAC count goes down. Now with LPS they go up. You see, you get a proliferation of alveolar MACs. 4OI could repress that proliferation, so we do think it can impact on macrophage proliferation in tissues. That's a work in progress, of course, but still there are effects on tissue resident macrophages as well.
Okay, thank you.
I think next question from Wei. Wei, go ahead.
Yes, this is Wei Li from NIH. Fabulous
Hi,
talk, Luke, as always.
Thank you.
My question is, could you comment on the different effect of DMF and dimethylmalonate?
Yeah.
Are they both acting through inhibition of succinate dehydrogenase, or they have any differences between the two?
Yep, that's a great question, right? Now, DMM is quite specific, as you know, for an SDH inhibitor. That's its main target, it seems. I mean, there are one or two other targets. DMF doesn't affect SDH, actually, we don't think at all. And its targets are these cysteine reactive proteins, basically, you know.
I see.
So it's quite distinct. Yeah, you might think there will be overlap, but and again, that tells us about specificity, doesn't it, in a way, that these metabolites can have quite specific effects.
Right.
No effect on SDH. Now, you might see DMF affecting OXPHOS, though, because it will have other targets that impact on mitochondrial function, you know.
Right.
That's a different thing, but there's no effect on SDH.
Cool. And did you ever try to use both and see if this
No, that's another good.
additive?
Geez, I got some great ideas from this. It's great, John. That's another good idea, to be honest. Yeah, it'd be interesting to combine them, wouldn't it? You may remember I worked with DMM. We got a big boost
Right.
and with DMM.
Right.
Great.
Right.
That's really good in vivo as well, by the way.
Yeah.
So it'd be very nice if you get that combination of an IL-10 booster combined.
Right. Exactly.
For now, you might get a really good effect there.
Yeah.
Yeah.
Well, great. Thank you.
Thanks, mate.
Okay, so I guess that's pretty much wrap up the talk. Thank you so much. You see, there's a lot of questions. I believe that if you guys have any of the following question, you can email Luke about your questions. And I think, there's a lot of interest in the field. But thank you so much for the talk, and I really appreciate your contribution for our seminar series. And then hopefully you have a good time in Canada and stay safe.
Yes, hopefully if I get there.
Yeah.
You never know.
I think it's going to be an adventure on the plane, and then
I think there is going to be a big adventure. That's right.
It will be. It will be.
I must take some DMF before I go, maybe.
Not such a bad idea. But yeah, you had a
Right.
great time in Canada.
Lovely. Thanks very much,
Okay.
John. Thanks for the invitation. And thanks for all
Absolutely,
the questions.
absolutely. Hopefully see you soon.
See you soon indeed. Yeah, all the very best. Thanks, everybody.
Bye bye, everyone.
Bye bye.